Now, the interview is part of our Wuji AppTech Thought Leadership Series, Innovation That Matters, and we seek insight perspectives from key opinion leaders and industry executives such as yourself. Our series focuses on novel and newsworthy advances in bringing transformative medicines to patients, the impact of collaborations and partnerships, views on new technologies on the horizon, and final thoughts on the future outlook of our industry. And it's a pleasure for me to introduce and interview you, Robert, as a co-founder of Galera, and where you've served as Chief Operating Officer since 2015. The company has focused on aspects of radiotherapy treatment, looking at dismutase mimetics to reduce oral mucositis, and also for increasing the efficacy of radiotherapy. And so the first topic that I'd like to introduce to you is that of in today's world of cancer therapies, how does radiotherapy fit into these cancer treatments?
It's actually a key part of most patients' patient care cycle. In fact, the statistic you see out there, and our research into it seems to indicate that at least half of cancer patients will undergo radiation therapy sometime in their cancer care cycle. Depending on the cancer, it can be curative intention, for example, with head and neck cancer, locally advanced head and neck cancer. 70, 80, 90% of patients can essentially be cured using radiation. In other cases, it's used to slow the disease or to control symptoms.
So when you're looking at these cancer treatments, they can be particularly harsh on patients, including poor efficacy and as well as side effects. So the approach that you've taken at Galera focuses on small molecule dismutase mimetics. Can you describe some of the science behind that?
Oh, absolutely, Rich. The science behind the dismutase mimetics is actually about improving both sides of that equation that you described as how it's hard on the patients. The toxicity. Toxicity can be one of the most limiting and debilitating things about cancer radiotherapy. For example, in our lead indication with our lead candidate, we're going to reduce, or we're looking to reduce the severe oral mucositis due to radiation therapy for head and neck cancer. The other side of that equation is the efficacy, as you described it, the anti-cancer efficacy of the radiation, where if we can improve that, you're also helping the patients greatly. And that looks at our locally advanced pancreatic cancer pilot study that hopefully we'll get a chance to talk about in a few minutes.
What's the inspiration of the approach, and then how is it unique relative to available therapies that are out there?
Right. Well, the inspiration is that there's a fundamental difference between normal cells and cancer cells in the way they deal with oxygen metabolism, if you will. Radiation produces large amounts of superoxide. And that superoxide, which you can basically think of as just regular oxygen, molecular oxygen with an extra electron, will rapidly react with all kinds of biological molecules and initiate normal tissue damage. It turns out that cancer cells are more resistant to the superoxide, whether produced by radiation or whatever the source is. So by removing superoxide, which is what our drugs do, we can protect normal tissue from the toxicity of radiation. On the other hand, what we do with that superoxide is convert it into hydrogen peroxide. And hydrogen peroxide, just as many of us have in our medicine cabinets, is a combination of two hydrogens and two oxygens. And cancer cells don't deal with that well. So cancer cells are much more sensitive to hydrogen peroxide than normal cells are. So the flip of the situation with superoxide. So what you're looking at is our drugs with one mechanism can remove the superoxide, protecting the normal tissue, converting it into hydrogen peroxide, which at high enough levels can increase the anti-cancer efficacy. It can be toxic to cancer cells.
So how do you sort of explained a moment ago how the mechanism basically selects for cancer tissue over normal tissue. Now, GC4419 received breakthrough status by the FDA for the reduction of oral mucositis. Now, how does this fulfill the needs for head and neck cancer patients?
So as I mentioned a couple of minutes ago, in the head and neck cancer setting, if patients are identified early enough with simply local disease, radiation and or surgery can be essentially curative for those patients. It's one of those cancers where we've made great progress in the past few years in terms of what we can offer patients. In approximately 40,000 patients a year in the U.S., the head and neck cancer patient receives 35 doses, small doses of radiation in combination with a systemic drug, cisplatin. And that can lead to essentially cures, at 1 year, 2 years, 80, 90% of those patients have no cancer. And so for the head and neck cancer population, what the real need is the severe side effects that come with this. So severe oral mucositis, which is the inability to eat or drink because of the oral mucositis, is the big need there. And so we've gone after that first with avasopasem now in the phase 3 Roman study, because for them it's the pain and suffering. They quite likely may receive years or may be cured of their cancer through the radiation therapy, but it produces this blistering of the inside of the mouth that by the time you get to what's known as severe oral mucositis, which affects 70% of them, 70% of the patients who receive the treatment will develop severe oral mucositis. At grade 3, they can't eat solid food. At grade 4, they can't even drink. And the pain, one oral mucositis and head and neck cancer survivor described it as like eating glass every day. So it's a really horrific complication that is what we've gone after, because we know that superoxide, which we remove, drives the initiation of that severe oral mucositis. And as you said, based on our phase 2B results, the FDA granted us breakthrough designation for severe oral mucositis.
That's really terrific news. Hopefully we'll have a good cure for that as well going into the future. What about your second drug? You were talking about enhancing the efficacy of radiation therapy. Can you describe that a little bit better for us, please?
Right. Well, GC4711, it doesn't have a snappy name like avasopasem yet. GC4711 is our drug for combination with radiation, and not just any radiation. So I mentioned a few minutes ago that the head and neck cancer patients typically receive 35 days of radiation at a small dose, what the radiation oncologists call 2 gray. So that's a measure of the amount of radiation given 35 times. And at 2 gray, you essentially make 2 gray worth of superoxide, and we can remove that and protect the oral mucosa, for example, and turn that into hydrogen peroxide, about 2 gray worth of hydrogen peroxide, which probably isn't enough to be incrementally toxic to the cancer cell. But if you look at what's now known as SBRT, which is fewer, larger doses of radiation. So, for example, 10 gray, so 5 times as much radiation per day given for 5 days, as is used in lung cancer or in pancreatic cancer. Well, now you make 5 times more superoxide. We turn that into 5 times more hydrogen peroxide. And in preclinical models, what we showed was that leads to a massive synergy. In fact, we were able to cure the mice of their tumors with the combination of our drug and that type of radiation regimen. So with 4711, we've optimized it to go after that combination with SBRT radiation. And in fact, before we even brought 4711 into phase 2 studies, we were studying it in phase 1 studies in healthy volunteers. And meanwhile, we initiated a pilot study using avasopasem because they have the same mechanism of action, similar pharmacokinetics, but 4711 is our SBRT optimized candidate. So we've actually recently announced that we completed enrollment of our pilot study with avasopasem and SBRT in locally advanced pancreatic cancer. Importantly, that's a randomized study. So it's a pilot study, but it's against a placebo control arm. We've completed enrollment, and in fact, this morning we announced that we've received late breaker status for presentation at ASTRO at the end of October. So we're very excited about that. We've already also announced that we're going to start a study of 4711 in combination with SBRT in non-small cell lung cancer. So you may remember that those are two of the areas where SBRT is currently already used. In fact, in lung cancer, some patients are now treated with SBRT instead of with surgery. So there aren't a lot of drug-based solutions to help the radiation oncologist, to help the cancer patient being treated with radiotherapy. So we really would represent one of the first, assuming we're approved, with avasopasem for severe oral mucositis, and then potentially for other indications. For example, we have a study now ongoing in radiation esophagitis, and you can think of other places where radiation toxicity is the big problem. And then if we're able to also get 4711 approved for combination with SBRT to improve anti-cancer efficacy, well, the radiation oncologists have been looking for something to make this kind of transformational change in their practice. And we think that really tailors itself for something that an emerging company such as Galera can launch and market ourselves. We're the experts on what's going on. We're probably the best to help our partners in the radiation oncology field learn how to use it and use it to help patients.
Thanks. Now, along the way, collaborations usually play a very key role in today's environment of drug development, especially in getting these medicines, these transformative medicines to the patients quicker. How did they impact the Galera's objectives? And maybe you can highlight that with an example.
Yeah. Well, part of our selection process for indications and drugs is the interest of key opinion leaders and of leading academic researchers in an indication we go after. So we wouldn't really pick up a field unless we had already vetted it and identified as a key unmet need where there was understanding that our solutions could play a role in that. So I think as you look at, for example, severe oral mucositis, that grew out of outside collaborations. And now as you look at our combinations with SBRT for anti-cancer efficacy, that's grown out of work with, for example, University of Texas Southwestern, which has been leading our discovery efforts in the lung cancer space I mentioned, and with MD Anderson, which has really been the cornerstone and was our first site for our locally advanced pancreatic cancer pilot study that we're excited about, seeing the top line results very shortly here. So academic collaborations are key, but so are corporate partners. And one place in particular, and I know this is near and dear to Wu Xi's heart, is the space of manufacturing and drug substance, drug product manufacturing. Because as an emerging company, you can do the biology, you can outsource the toxicology, but it really has to be a partnership to develop the synthesis of drugs, convert those into GMP processes, to formulate them into drug products. So I think a key collaboration for an emerging company like Galera are the ones we strike up with our contract manufacturers. I like to say that CROs, contract manufacturers, are kind of like your family. They can make mistakes. It's what they do after that. And I think that's the key thing I look for is the people we work with, we want them to be good, and we want them to be good at working even when there are things to figure out.
Thank you very much. Let's turn our attention to lessons learned. Now, Galera is developing mechanistically novel mechanistically novel mimetics. What have been the challenges along the way, and how are they solved, and what have been the lessons learned?
Well, Rich, you just nailed the number one challenge, which is we are a virtually unique class of drug. If you look at drugs out there, I would guess that 95+% are either inhibitors, which is the bulk of that 95%, or they might be agonists, things that increase the activity of a target. Whereas we're actually a mimetic of an existing enzyme. So the closest analog you might think of is insulin, which is a product. And so there's synthetic insulins that are given. And in many ways, that's a good parallel. So that's been one of the biggest challenges is just people understanding we're not inhibiting superoxide dismutase. We're not increasing its activity. We are a mimetic of its activity, which allows us to augment it in settings such as radiation, where there are superoxide dismutase enzymes, actually 3 of them, in every one of our bodies. But when you get radiation, it produces more superoxide than your native enzymes are capable of dealing with. So we enter, in that setting, we enter in with an augmentation that relieves that that burden of excess superoxide. I think the other piece that's novel there is that we are a class of drugs that includes manganese as the active center of this molecule, and so that's also a unique motif for regulatory affairs and even for contract manufacturers. That's one of the things that's distinguished the contract manufacturers that we've collaborated with, continued to work with, is their ability to wrap their heads around a fairly unique chemistry. It's not like most medicinal chemistry.
Right. When you take a look at near-term and longer-term outlook, how does the current environment either shape or reshape Galera's near-term objectives, and how does it affect its longer-term positioning in the industry?
Well, I think it's important to understand that no matter what the underlying environment is, either health-wise or otherwise, people continue to get cancer. We've been fighting a war on cancer for many decades, and it is still a battle that is very much not yet won. So people continue to get cancer, and they can't wait for various externalities to resolve themselves. So our business has actually continued pretty much as is in the current situation. We did, as I think we announced, delay or rather had some uncertainty around the starting time of a European study for severe oral mucositis. But actually, we found that we were able to meet guidance on that and start that study in the first half of this year. And actually, we found that it is true that cancer patients continue to need their therapy no matter what the outside influences are. So we continue with all of our clinical studies enrolling nicely.
Thanks. Are there plans to extend the application of the technology to other indications? And then one last question along that line would be, any kinds of improvements in the technology that you can foresee or plan?
Absolutely to both those questions, Rich. So as I mentioned, oral mucositis is mucositis of the mouth. We've announced and have begun a study in radiation esophagitis, which is really mucositis of the esophagus. The reason we started with oral mucositis, with severe oral mucositis, is because you can observe the patient's mouth. You can see they have ulcers. It's relatively straightforward to assess. Esophagitis is somewhat more difficult to assess. It relies more on symptomatic kind of evaluation, but it can actually be significantly more deadly. An unfortunate number of patients with radiation esophagitis may acutely die because of bleeding or actual rupture of the esophagus, so it's a big problem. And so you could imagine that there are other mucositis all the way down the GI tract. As we've also published on, we've demonstrated that we're able to reduce the damage to lung tissue due to radiation therapy. And so, in fact, our study with SBRT in non-small cell lung cancer, in addition to looking at tumor outcomes, will look at the development or hopefully the reduction in development of pneumonitis, which is essentially radiation damage to the lungs. So in terms of improvements to the pipeline, we have a number of molecules in discovery as well as other indications that we're looking at that address specific needs, but I don't think we've gone into any kind of public detail on what those are.
Okay, well, thank you.
We're looking to transform radiotherapy. And by that, as we've been talking about here, it's both making the radiation therapy more tolerable for the patient and making it less tolerable, making it toxic for the cancer. In doing that, we'll be touching the lives of hundreds of thousands of patients, we believe, going forward, who hopefully we can not only make their lives better, but extend their lives. It's why we get up in the morning. It's why throughout the current situation, we've been in our research labs moving these forward. It's why we're excited to see our pilot study results at the end of October, and why we're really excited to see our phase 3 results, in the second half of next year.
Thank you so much for a wonderful and insightful perspectives on this emerging area. Thank you very much, Robert. We really appreciate it. Thank you. Hey,
take care.
Bye-bye. Thank you. Bye-bye.